Welcome to Forschungszentrum Jülich, which is the biggest national research center in Germany, located between Aachen and Cologne in the western part of Germany. Here about 5,900 people are working together to advance science in various directions of domains. For example, in materials, in energy or neuroscience. Here we are located in the Jülich Super Computing Center, which provides supercomputer resources for the scientific community in Europe and also in Germany. But not only the provision of computer time is essential, we also organize workshops, tutorials, and are very much engaged in education of young researchers and students. Computers are nowadays influencing society, economy, and technology to a large extent. But not only these three fields are influenced, but also scientific research is influenced. So in science, we do actually a lot of experiments now on computers in a virtual environment. So meaning that we do numerical experiments and performing experiments according to a model which we construct beforehand and then analyzing it in terms of numerical algorithms. So these numerical algorithms and numerical models are used then actually either to predict new phenomena or also to interpret experiments which run in laboratories. And so that we can use the data which are generated by the experiments to go in very much detail in the experiment and to analyze what's going on and to understand actually what is the behavior of the matter and what can we say about nature in this respect. The EUSMI facility now offers a computer here at JSC, the so-called JURECA machine, which you see here as during the installation, which is composed of more than 40,000 compute cores, meaning that we have actually independent compute unities which can do work in parallel together, meaning in principle that you can speed up a given problem by a factor of 40,000 in principle and finish your research in a given amount of time. For example, we have 16,000 cores and working on 24 hours a day would mean that an experiment what you would do on a serial machine would take about 40 years compared with 1 day on this machine.
Life begins when the sperm meets the egg. Using the supercomputing facilities of EUSMI, we were able to model swimming of sperm cells. And in the same way as you observe for sperm cells swimming through in the microscope, we see that the sperm swims through the fluid, reaches the surface, turns around and swims on a circular path at the surface. Now comes the advantage of using computer simulations. Of course, we've just observed what we see in the experiment as well. However, in the simulations, we know exactly where the sperm is when, like a perfect microscope. But furthermore, can we turn on and off different aspects of the interactions? For example, we turned off hydrodynamics, that is fluid flow. And in this way, we were able to show that it's actually the fluid flow that keeps the sperm cell at the surface. Two effects contribute to this surface adhesion. For one, the sperm cell sucks in fluid from the side. And like a vacuum cleaner, this sucks the cell towards the surface. And as a second effect, fluid gets pushed out at the back, which sort of turns it away from the surface. In this way, it's pointing towards the surface and remains there. Moving to a more complex geometry, we simulated sperm cells moving in microfluidic channels. Again, the sperm cell adheres to the sidewall thanks to the hydrodynamic interactions. With the computer simulations, we were able to study how the sperm cell, when it reaches the corner, scatters at the corner. And we can predict how the scattering angle depends on the beat pattern of the sperm. This may, in the future, allow us to separate sperm according to their beating pattern. Cilia are one of the very conserved structures throughout eukaryotic life. From animals to plants, from the unicellular paramecium all the way to the human lung or the human brain, cilia propel fluid across the cellular surface. These hair-like appendages of this cell beat in this wind-like fashion, as you see in the background here. There is the elongated, stretched out power stroke that moves the fluid across the cellular surface, followed by a bend closer to the surface recovery stroke, which brings the cilium back in its original position. These cilia, however, do not beat all in perfect synchrony, as you would expect, for example, from a rowing boat. Neither do they beat completely in a random, chaotic fashion. Instead, they beat in this wonderful wave-like pattern that you see here in the back, similar to maybe a wheat field in the wind. Using computer simulations, we were able to show that hydrodynamic interactions, i.e. forces mediated by fluid flow, lead to this self-organized synchronization of cilia beating. Furthermore, we could show that this is highly beneficial for the cells. This metachronal coordination not only leads to roughly twice the speed of fluid flow, it furthermore enhances efficiency by roughly a factor 10. Volvox are a paradigm example of a ciliated microswimmer. These spherical algae swim by a carpet of cilia on their surface. We modeled a spherical microswimmer covered by the same cilia I presented before. And we do indeed see that this microswimmer nicely swims forward. However, how exactly the beating pattern of the cilia influences the swimming speed of the algae, or what is the optimal arrangement of cilia on the surface, remains a mystery to be solved.
Within the EUSMI project, we offer supercomputer time on the JURECA machine here at JSC in Jülich. This is open for academia and industrial users. To support the porting and optimization of codes and to facilitate the access of the supercomputing facility, we offer support by our simulation laboratories. Welcome to EUSMI.